Here I'm going to give another example using some real-life images. So here, for example, this one. All right, so this is one of the examples of multiple cells in the same field of view. And if you want to the statistic based on the single each cell, then probably surface and the spot is not really easy to get this result. So in this case, we can try to do the cell. And in this case, we don't have the nucleus staining, so we can just use the third option, detect cell and the vesicles. So let's say I want to get a result of how many vesicles in each cell, and I want to filter out those cells with relative large number of the vesicles in this field of view. I want to get a statistic. So here, again, we need to detect the cell type. Here, I'm still using the cytoplasm staining. So here I use green channel and enable the smoothing. Also the background subtraction. So here I need to give Imaris the size of these cells is about 8 micron. Yeah, it's about, I can use 8, I guess. Okay, so here again, I can do the volume display like this and try to make a better thresholding. So you can see due to the staining, there are some small holes. Then if you want to get rid of this, you can probably go back and maybe give a slightly larger smoothing to see if it will be improved, like this, 0.3. Like this. So basically the hole has been filled, but then you will see some connection between these close together cells. Then here we can enable the options because we don't have the nucleus staining, so we need to split by C points. So I would say 8, the cell size. Okay. So here I try to lower the threshold limit to include all the detection like this. In the meantime, I can see there are some cells close to the boundary, which is also detected. But if you don't want this included in the detection, we can get rid of these cells which is close to the boundary in the later step. Okay, so now I have this separated different cells, so I can remove the small ones first by using this filter. And also in this step, I can add another filter, which I should use cell, I think this is the distance from image border. So now I can remove those very close to the image border once, because they are not a complete cell, so I want to get rid of them from my result. So I can do these multiple filters to remove this from my final result. Okay, so for the vesicles, now I can turn off the cell display and hide the green channel. So now only red channel enabled. And the same, I need to give the diameter of this structure. So 0.5 maybe. Now here I'm just going to use the same size because I don't care about the size. I just want to see the numbers. So Okay, so I can lower the threshold to have more detection. So here, the vesicle, it only gives you the vesicles inside your cell detection. So as you can see here, there are some bright spots, bright vesicles, but it's not detected whenever whenever the range of your vesicle detection, it will still not be displayed because it is not inside the cells. So only the vesicles inside the cell will be shown here. So sometimes you need to check your cell detection to make sure it is including the whole cell volume. Sometimes you will try to adjust this threshold, but you can still not unable to see this detection. Sometimes it's because the cell volume is not including the whole stemming. So you have to go back to the detection cell detection step, and check your cell detection volume. Okay, so now I finish my detection, and here again, cells. If I go to statistic and cell number of vesicles, I can already see these numbers. But if I want to display it in a more, I want to visualize it. So I can go to this color tab, and switch to cells. And this time, instead of using the base color, I can probably use the statistic code. And I want to color code based on the number of vesicles. Right, so in this case, I can also turn on the transparency. So now you can see the ones with largest number of the vesicles will be coded into the red color, which is this one. This one probably with 89 vesicles. Yeah, and some other cells with less number of vesicles will be color coded into purple or gray color. So now you can have this visualization of your results. Then you can do a snapshot or do the animation, and of course you can export this whole table for further statistic. So this is one good example of how you can use cells to get this result in an easier way.